Cell migration plays a crucial role in developmental biology and in tissue engineering. So, we will try to understand what cell migration is, why cells migrate, what are the roles of this cell migration and so on. Okay. This is in general a very important process for both unicellular and multicellular organisms. For a unicellular organism, it is usually for finding food. So, the cells migrate, they usually swim through a fluid, try to find if there is nutrients which they can use, right. So, this or they can also crawl on a surface as an amoeba would do. Multicellular organism also does cell migration, but for more complicated processes. It is not just for finding food, it could be for tumor invasion, metastasis, embryogenesis, angiogenesis or immune responses and so on. So, if you remember when we looked at wound healing, so the aspect which we talked about was migration of inflammatory cells, phagocytes, macrophages and so on, to see how they actually come to the site and so on. So, migration plays a crucial role in some of these aspects. So, the speed and the pattern of migration is actually determined by the type of cell and also the chemicals present in the environment. So, some of these molecules can act as triggers or signals for directing cell migration. So, the fundamental mechanism for forming structures within embryo is cell migration, right. So, think about it, you only have one cell to start with, it starts dividing. If the cells just keep dividing, you would just have a blob of cells, right. You are not going to get defined shapes. So, what is going to happen is these cells are going to produce matrices and they are going to migrate to form different tissues, right. So, different organs and tissues, and so because of this during embryogenesis cell migration is very crucial. It is also very well controlled, and that is why you end up with two eyes on the place where they are and a nose on where it is, right. So, it is a very regulated process, and this ensures that cell migration is controlled. So, this requires cell migration to be controlled. So, development of tissue structure and cell migration are interdependent, right. So, only when you have the cells migrating to a particular area, the tissues can start developing. So, people have used magnetically labeled oligodendrocytes progenitors and monitored them using MRI to show that the cells can actually cover a region of greater than 8 millimeter in size during the first 10 days when you inject it to a spinal cord. So, when you are talking about tissue engineering applications, cell migration can be very important because if cell migration is going to be there, you might have host cells coming to the tissue construct and integrating very nicely with the cells. And also in case of angiogenesis, cell migration is required for vascularization to happen. However, it is not desirable if cell migration can cause side effects such as loss of function at the transplantation site. So, basically if you have a cell seeded scaffold and these cells are just going to migrate away, then you would have a problem, right. So, you would want to ensure that the cell migration is optimized. So, can you think of a way you would minimize this type of loss of cells from a cell seeded scaffold? If they adhere more strongly, if they prefer to adhere to the scaffold compared to what is present surrounding it, then they will probably not migrate away, right. So, optimizing the scaffold for cell adhesion can actually help in controlling cell migration. So, these are some of the cells which migrate very commonly, and these are the roles when it comes to the migration. So, neutrophils migrate for phagocytosis, lymphocytes for destruction of infected cells, macrophages and for antigen presentation and in response to antigen presentation, and endothelial cells to create angiogenesis, epidermal cells and fibroblasts during the process of wound healing, tumor cells during metastasis they will migrate, and neurons and axons during the development and regeneration of nervous tissues, and embryonic cells migrate during embryogenesis. Right. So, if you are looking to study migration of cells, how would you go about designing an experiment to study cell migration. So, no, migration does not always happen through the blood vessels. It's not that the cells which are adhered to a surface actually enter into the blood vessel and they are carried to some place. It can happen during metastasis in some cases, but migration for it to move. So, it cannot, it has to come from somewhere to the blood vessel if it is even going to do that, right. Cell migration would have to happen, can happen on the surface as well. Right. And see, many times cells are proliferating, they also have to migrate, right, otherwise everything will just be forming a blob in that. So, what you basically do is you fluorescent label the cells, and these cells will be tracked using time lapse microscopy or conventional microscopy. Confocal can be used, but confocal is usually more complicated. You can actually use it if you tag it with fluorescence, you can use simple microscopy techniques. And what you would do is you will see where the cells you seeded are and where it actually is looked at, okay, how far it has gone. You can use sectioning to see how far it has gone and so on. Okay. So, what is the commonly used fluorescent molecules for these studies?
Can methylene blue be used?
Not really. What is one common fluorescent molecule which you know for biological studies?
Something called fluorescein.
Fluorescein diacetate. Fluorescein diacetate is to stain live cells, but yeah, you may be able to use it, but the most common molecule which is used is GFP. Okay. So, lymphocytes and other phagocytic cells can also be tracked using MRI, because they will ingest these superparamagnetic particles, right. So, these are phagocytic cells, they gobble up stuff, right. So, if you put a nanoparticle there, it will eat it up, and you can track them using MRI or other microscopy techniques. So, when you are talking about cell migration experiments, what you look at is to introduce some kind of genetic markers to the cells that can actually be tracked. So, time lapse techniques can permit quantitative analysis of individual cell movements, and you might have seen these videos. So, in some publications they usually have these time lapse videos. I do not know if you have, none of you might have taken my immunology, right? Did anybody take immunology here? Okay, so you did? Okay, so I am pretty sure Dr. Vani would have shown that video where a neutrophil runs behind a bacteria. So, a neutrophil basically, it's a time lapse video actually, it's not that it's going at that pace, it's about a 30 seconds to 60-second video, and you would be able to see neutrophil chasing a bacteria to eat it. And so, you can look up such videos. These are actually time lapse videos which is taken over a long period of time, and you would many a times you would actually see a clock which is running much faster than the actual time to show you what is the real time frame. So, the most common migration assays which are used actually expose cells to very simple systems compared to in vivo setup, right. So, in vivo the tissue is much more complex, whereas the migration assay, if you are going to culture it on a plate and look at the migration, it is going to be very simple. So, can you think of a common limitation which you would experience with such systems?
If you do it on a plate, then it will be 2-dimensional versus 3D as it will be.
Yeah, that is usually the major problem, right. So, most of the cell culture which is done in a 2D setup, like only when people work on tissues they start looking at 3D setups. So, that is important, because when you have a 3D environment, the migration directions also increase. So, you might have to observe things from different angles. So, when you are talking about 3D environment for migration, basically what you do is similar to what Karan was alluding to. So, you basically have a collagen gel on which the cells are seeded, and you follow the cell infiltration into the gel by following the leading front of the cell population. See, not all cells are going to migrate at the same rate. So, you might have a lead cell which is what you would track to see what is the max, how it is actually moving. So, this gives important information about cell migration. However, this experimental system would have an important limitation. Can you think of any two limitations?
cells.
Yeah, any migrating cells, any cell migration.
Because of its 3D motion, the cell goes out of the focus.
Not necessarily. You can, those are, I am not talking about observational problems, I am talking about a design problem in the experiment.
The scaffold's design will impede how it moves, migrating.
But that is what is supposed to happen, right? The ECM is going to have its own
Design.
barriers. So, okay. So I will tell you. So one thing is it actually is a very long process. So it can take days for it to infiltrate, and monitoring it and maintaining the correct environment, making sure the cells do not die, are all quite big challenges when it comes to performing these experiments. And the other issue is more of a proper design problem, which is the leading front distance only gives information about the fastest moving cell. It is not the average cell population which you are looking at. So whenever you are looking to collect data, you are trying to get something representative of the sample. This will not be truly representative of the sample. This will only represent the fastest cell.
Sir, how are these fastest ones chosen? Like
No, because that is the leading edge you would be able to observe, right. When you observe it in a microscope, you will just be seeing the leading front as it is moving, that is all. So, see, if you were to have a fluorescence tagged thing and you are observing it in a microscope, you just look at the farthest cell from the starting point, that is all you are looking for. So, this is how a cell which is migrating would look like. So, what you see here is the front region, which is called the lamellipodium or the leading edge, and the rear region is the uropodium, which is seen in the back. So, what happens during this time is the cells can actually what happens during this time is the cells which are adhered basically start moving, and they go and adhere to some other surface, and then they get detached from the previous site. Okay. So, once they have done that, they have actually moved one position. So, the periods when the cell is moving forward in a straight line without turning is called as the persistence time. So, when you are talking about a cell migrating, the cells are not going to always move in random directions, right. So, they will continue in one direction for a while, and then they may change directions as well. So, this time frame in which it actually moves in one direction is called the persistence time, and this persistence time can be altered based on having chemoattractants and so on. So, if you have a chemoattractant, then the cells are going to try and migrate towards that attractant, and in that case the persistence time will be longer. Otherwise, if it is going to be uniform distribution of the chemoattractant, the persistence time will be much shorter. Okay. The cell movement itself is initiated by active membrane protrusion in the lamellipodial region. So, in case of fibroblasts, the leading edge of the cell protrudes and retracts in a cyclic fashion. So, basically the peak velocity it can reach is about 50 to 60 nanometers per second, and it averages a net speed of 5.5 nanometers a second. So, that is the rate at which they usually move. So, during this cell movement, new cell adhesion sites have to be formed, right. Only then the cells can leave the previous adhesion site and go to the new adhesion site. So, these are some of the cells, their persistence time, speeds, and their motility coefficients. So, what you can see is some of the cells which are very motile would have lesser persistence time, like your neutrophils or macrophages have lesser persistence times. So, especially neutrophils have significantly lesser persistence times because they just have to figure things out in the region where they are, right. So, they cannot just keep moving in one direction, whereas if you were to look at endothelial cells, they are actually not as migratory as these cells. So, they will have a much higher persistence time, and whereas if you are looking at speeds, obviously the cells with lesser persistence times end up having higher speeds and so on. So, this is the process which describes cell migration. So, this is a repetitive process of lamellipodial searching for a new adhesion site. So, the new front edge which is coming out forming will basically have to go and adhere to a new surface, and once it is adhered to a new surface, the uropodial region has to get detached and it moves to the new site. For this to happen, what would have to happen is the cell would have to strongly adhere to the new site. So, the initial step is for the cell to stretch, to figure out what is the right place to adhere, and once it adheres there is now a tension in this cell, right. This is because the cell is stretched. So, this tension could cause it to either move forward or move backward, depending on whichever site has the higher adhesion properties, higher affinity. So, if the cells have higher affinity to the new site, it will move forward. If it has higher affinity to the old site, it will move backwards. So, this is something which you can even simply test as well, right. So, like I do not know what are these jelly kind of clay which is called, like now they are slime, yeah. So, you can play with that and you will be able to emulate something like this. So, if you stick it strongly at one site and you stretch it out and place it in another site, it will either come to this site or go to the other site based on how strong your adhesion is. Okay. So, let us look at some of the different adhesion properties of the surfaces and how that would affect your cell migration. So, what would happen to the cell migration speed if your substrate has very high adhesive properties?
The initial site or
The surface in general has very high adhesion. So, because it is not like I am not going to look at one cell, right. If I am preparing a scaffold with very good cell adhesion properties, then how do I expect the migration speeds to be?
if it's high, it will be slow.
It will be slow because it is already adhered strongly, it might not want to go to another site. Okay. What if it is very low?
Then though it will lose migration will be very fast.
It depends. I mean, if it's easier for it to migrate, but that doesn't necessarily mean you want.
I think it might be faster.
If it is not adhered to the site, it means that it does not really like the site, right. So, it might want to migrate as well.
But it's not getting the right grip to pull itself.
So, that would be the problem. So, in the first case what will happen is it will not be able to detach itself from the first site, whereas in the second case it will not have a position where it can actually attach and move. So, in both cases the migration rate will be very low. So, when it is medium, that is when you would have high migration rate, and as I have always said, biology always has some optimal values, right. So, high, low are never good, something in between is where you are looking for. Okay. And these predictions have actually been confirmed experimentally as well. So, when you are talking about cell migration, there are two major types of migration. You have a random migration. So, this is observed for when you look at it for a sufficiently long period of time. So, what would happen is if you are going to observe, see we looked at the persistence times, right. So, we said that the persistence time for endothelial cells is 300 minutes based on the table. So, if my period of observation is only 150 minutes, then I am going to think that the cells are moving only in one direction, right. And if I am going to observe only one cell as well, like smaller number of cells, that is going to happen. If I were to observe a very large number of cells for 3000 minutes, then what is going to happen is different cells are going to keep moving in different directions. So, it will look clear that it is a random migration which is happening, assuming there is no signal which is actually directing the migration. Directed migrations happen when it comes to cells actually responding to their environment. So, the rate of migration will depend on the composition of the local environment, whether the receptors are present and what soluble factors or surface bound factors are present. So, if the ligands are present on the surface, then the cells will migrate towards that and so on. So, if the environment contains uniform concentration, then the migration becomes random, right. So, if you have one region which is rich in these ligands, then you might have a directed migration. So, if spatial variations are present, the cells are actually capable of detecting these gradients and changing their migration pattern towards these attractants. So, this process of moving in response to some gradient is called as taxis, and it is critical for regulation of any biological event. that involves cell movement, because otherwise if cell movement is always going to be random, then you would have very little control over tissue development or anything, right. So, this directed migration ensures that this is happening. Okay, so, in case of random migration, I had also put some term called Brownian motion. So, what is Brownian motion? Movement of particles in? Okay. Why would it happen? It has its own energy, kinetic and internal energies, which makes it move, and as it collides they change directions, right. So, that is why it is random motion. So, that is why it is Brownian motion. Okay. So, if you observe a random migration of cells for a long period of time and you track the cells, it will look like it is a Brownian motion because it will be quite random. So, basically Brownian motion is a solute particle moves as results of collisions and their own energies. So, the momentum is transferred from one particle to the another during the collisions. Particles move with a velocity, I do not know, v prime has actually moved. So, they can move with a velocity of v dash until a collision causes it to change directions and then move with a new velocity. Let me just fix this. Okay. So, they can move with a particular velocity until some collision causes it to change directions. So, otherwise it will keep persisting in that is your persistence time, right. So, only when there is a collision there is going to be change in direction. So, that will affect your speed as well, velocity as well. So, there could be momentum which is lost or momentum gained based on the velocities of these two molecules, velocities and mass of these two particles which are colliding. So, collisions occur from all directions with equal probability, that is what causes this kind of a random motion. If it is going to be in one particular direction, then you are going to have, you are not going to have a random movement, right. So, in case of Brownian motion, the time between these collisions will depend on the density of the molecules, right. So, if there is going to be a higher concentration of the solute molecule, there is going to be a higher chance of collision and so on. So, the average time between collisions can be estimated from the average distance the particle travels between collisions, which is called as the mean free path, and the average speed at which these are moving. So, the movement of a collection of Brownian particles is typically characterized by the rate of increase in the mean squared displacement over a period of observation. So, usually this time frame for observation is much larger than the mean free path interval. So, similarly if you were to draw analogy to our system, your period of observation has to be much larger than the persistence time. If you are so that is why I was saying if you are looking at 300 minutes and have observed it only for 150 minutes, it is not going to work. So, the persistence time has to be much smaller than the period of observation for you to understand random migration. So, this behavior can be characterized by a diffusion coefficient d, which can be given by the Stokes-Einstein equation. Okay. So, we talked about how random migration can look like Brownian motion and what Brownian motion is, but are the mechanisms similar? Okay. So, what would be the difference?
I don't think there would be collision between cells.
So, it is not that the momentum transfer is happening causing the change in directions, but change in directions is happening because of adhesive properties and other things, right. And the other thing is the cells do not, this change in direction is not instantaneous. In case of molecules colliding, you are going to keep coming boom and then go different ways. It is not this way, the cell migration is going, cell change in direction is going to be very slow. Okay. So, and also that is because it is not a transfer of momentum, but it is a sequence of significant number of events which include attachment to a new surface, a contraction of the intracellular fiber system and detachment from the previous surface. So, dead or fixed cells can also move because of Brownian motion. So, they will not migrate on the surface. So, those would just be particles, right, those will be particles in a suspension. So, neutrophils can actually migrate through a collagen gel 20 times faster than you would expect them to diffuse in water. So, although collagen is a much more viscous system than water. showing you that it is not that it is going to be dependent on the momentums, but it has mechanisms to make sure it reaches things faster. Right. So, neutrophils usually are trying to attack stuff when they have to migrate through the collagen matrix. So, they move very fast in that environment. So, although cell migration is similar to Brownian motion when you observe it for a long period of time, the striking difference would be that the cells can actually move in the same direction at a nearly constant speed when you observe it for relatively short intervals. So, the change in direction can occur randomly, and this is completely stochastic in nature, and it depends on the membrane adhesions and cytoskeletal contraction events, but substantial changes in direction occur only after small random perturbations have been accumulated. So, it is not like the cell which has been moving this way suddenly starts changing it and directions and moving the other way. It will then move slightly to another site before it completely changes directions, right. So, in spite of these differences, you can actually use mathematical studies based on Brownian motion for understanding cell migration, for random cell migration. Okay. So, in our department there is a Professor Murugan. I do not know if any of you have met him or not, but he actually works on not exactly on cell migration, but he works on aspects of protein migrations and protein folding using Brownian motion and so on. So, if you are interested in understanding these topics, he might be a person who might be able to help you with that. Okay. So, when you are talking about directed migration, there is actually spatial variations of factors which regulates cell migration, and this leads to cells detecting these gradients and changing their migration pattern. So, this process is called as taxis. So, there are different types of taxis which you would have heard of. Topotaxis is enhanced turning towards a stimulus. Orthotaxis is increased cell speed when the cell is already oriented towards the stimulus, and you have clinotaxis, which is decreased turning when the cell is oriented towards the stimulus, right. So, these are the 4 ways the cells can respond in the presence of a stimuli. Okay. And based on what stimuli actually is causing the migration, the process can be labeled as chemotaxis, haptotaxis or phototaxis. So, chemotaxis is when you have dissolved chemicals, some chemicals which are acting as the signaling molecules, and cell adhesion would cause haptotaxis, and light could be the trigger for phototaxis. Certain agents can cause chemokinesis, and which is a change in the kinetics of cells random migration that does not require a gradient, but will depend on the total concentration of an agent itself. Right. So, this is in response to a particular concentration, the cells will move, and again we use the same terms like orthokinesis and clinokinesis, and orthokinesis is variations in cell speed versus clinokinesis is variations in cell persistence time. So, the pattern for the random migration of cells, as we said, can be similar to Brownian motion if you observe it for a long period of time. However, if you look at it only for short periods of time, it will not be. So, you need to make sure that there is significant directional changes, so that you can actually look at it as random migration, as Brownian motion. Okay. So, however, cell migration is is actually a very good example of something called a persistent random walk. Okay. So, what happens is at short observation times, the cells seem to be persisting towards one direction, and for long observation times the cells are migrating in random, right. So, that is what is a persistent random walk. It represents both sides. Okay. So, in the simplest random walk, what happens is a particle is constrained to move along a one dimensional axis, and in a time interval tau, the particle can move a distance of delta depending on the speed in which it is moving, right. So, that is quite logical. So, at the end of this time period the particle will change its direction. So, this is the persistence time. So, at the end of the persistence time it will change directions and go into something else. So, the particle can actually go into left or right. So, in an unbiased random walk there is actually equal probability that it will go to either left or right. You understand? So, I am walking towards a certain direction. So, from here let us say I am walking like this, and if 3 steps are my persistence walk, and then I can turn left or right, and I can actually either way is fine. So, I do not really have a preference, and that would be an unbiased random walk. So, at each step a new direction is chosen randomly, and with the particle's history of the movement having no impact. That is quite logical, right. So, if you were to think about the cell, if once it gets detached from the previous site has no role to play in where the cells are moving towards. So, if many particles move based on these simple rules, at the end of the first time interval tau, half the particles will be at minus delta and half the particles will be at plus delta, right. So, if both have exactly equal, if all cells have exactly equal probability of moving left or right, then half would have moved left, half would have moved right. So, they would have moved to, so if center is 0 it would have moved to minus delta and the other would have moved to plus delta. So, the position of each particle after n time intervals can be given as x of n equals x of n minus 1 plus or minus delta, right. So, that would basically mean at n minus 1 this is my position, it could either be this or this, right. So, those are the positions I can be in after one step. So, from this equation you can actually calculate the mean particle position using this. So, basically you have just divided it by the number of particles, like summation of all the positions divided by the number of particles. Okay. So, now you need to simplify this equation. So, if you were to simplify this equation, can you think of what would happen to the second term?
Will it not get cancelled?
Why?
Because there's plus delta minus delta plus delta minus.
Yeah.
Will be plus half it.
So, half will be plus, half will be minus, so it goes to 0. Okay. So, this happens only in an unbiased random walk. Okay. So, if it both are if there are bias, then you cannot do the simplification. So, basically what happens is x of n equals x of n minus 1. So, what does this mean?
The mean position of the cells remains the same.
Yeah, it is independent of time. The position of the particle is actually not dependent directly on time. Right. So, all the average position is not changed, the particles are actually spread across the axis, right. It is not that the cells are not migrating. For every cell which is moving to the right, there is another cell moving to the left. So, its mean distance from its origin is different, they have increased, but if you were to add all cells that would even out. Okay. So, the mean squared displacement will give you the extent of spread. That is why you look at the squared value rather than just the actual value, because that will give you a 0. Okay. So, as the mean squared distance is 0 at time 0, the equation indicates that the mean squared distance increases with the number of steps, right. That is logical, right. So, as you have more steps you would have more distance covered and more spread. So, if the total time lapse is n tau, then you would, this equation basically, this equation would simplify to this. So, all I am doing is dividing it by n tau, right. So, the root mean squared of the displacement which is XRMS is given by this equation, and you have d equals delta square by 2t, and what people have shown is experimental results suggest that d for albumin is about 8 times 10 power minus 7 centimeter squared per second. D is the diffusion coefficient. Okay. So, as velocity between so as v is the velocity between the change in directions. During the persistence time they have a migration velocity, and that would be delta divided by tau. Delta is the distance covered, tau is the persistence time. So, that would be your velocity. So, delta can basically be estimated as 2d divided by Vx, and using this Vx of 600 centimeter per second for albumin, delta is 0.3 angstroms and tau is 10 power minus 12 seconds. So, that just gives you a feel for what this would be. So, whatever we looked at right now was a one dimensional random walk, right. So, it could only move left or right, it could not move front or back. From one point it only did left or right. So, in a two dimensional random walk you will also have front or back and other directions in the same thing. Okay. So, that would end up with an equation of square root of 4 dt, and a 3 dimensional random walk will give you square root of 6 dt. So, directed migration of cells in response to a chemical attractant, chemical agent is called as chemotaxis. So, this could be a chemical signaling molecule or a chemoattractant. A direction of migration is usually towards higher concentrations of chemoattractant. The strength of attraction will depend on the absolute concentration and the steepness of gradient, right. So, your concentration has to be high, and also the region where the cells are present, if the concentration is low, then there is going to be this gradient which will make it move faster. So, the activity of the chemoattractant increases with concentrations up to an optimal value, and then the activity starts decreasing. Okay. So, again chemotactic index is the term which is used to characterize the chemotactic movement. This is just an equation which describes it. So, so basically this is given as d divided by L path, where d is the straight line distance which is travelled through by the cell towards the attractant. So, if there is a chemoattractant, the cells will be moving towards that, and the distance which it persists towards it, that is called as is d, and L path is the total distance which is travelled by the cells, and p is the persistence time, and the correlation is t is obviously the time, and this is the correlation, and it can actually be simplified to get something like Ci equals d divided by L path. So, what do you think would be the chemotactic index for random migration? So, yeah, it would there is no real attraction, so it will keep moving all over the place. So, for a perfectly directed migration, 1, because your d will be L path, equal to L path, right. So, the persistent random walks with selected bias added to either the speed or the direction can be useful for studying the patterns of random migration in the presence of chemoattractants. Okay. So, that would be the directed migration, and simulations have actually been done to understand this. So, however, it is still not fully understood as to how cells recognize the presence of chemoattractants and what mechanism the cells use to determine the direction of the chemical gradient. So, but this is not understood in mammalian cells, it is very well understood in bacteria and other unicellular organisms. So, not all mammalian cells have been studied well enough to understand these factors. Okay. So, some of the factors which can regulate cell migration are soluble factors, ECM proteins, cell substrate interactions, even electrical fields have shown to impact cell migration. Okay. So, that gives an overview of what cell migration is, and the only aspects which we still have to talk about are reactors and how we use signals, and with that we will come to the conclusion of all the fundamentals related to tissue engineering. So, we have how many more classes left?